Yeah, so in today's class, we'll be talking about the mitosis in general, okay? And we'll be saying about the different phases in the mitosis, in the prophase and metaphase and all those phases. And this is regarding the MCDK cyclin complex. And this is regarding the activation of MCDK, okay? As we discussed over here that CDC2 and cyclin complex are important in the activation of the M phase. And there are these phosphorylation and phosphorylation sites on the CDC2 and cyclin complex, which helps in the activation of the M phase. So in the mitosis, generally these are the different events which occurs in the mitosis, and um, so first is the spindle body formation, duplication, and separation. Okay, spindle body formation is basically the formation of the centrosome and the microtubules organization. Okay, so centrosome is a is a pro, is a molecule which helps in the segregation. So here in this image, you can see that this is the centrosome, and uh, this centrosome is initially formed during the S phase of the cell cycle. Okay, during the synthesis phase, and generally the centrosome needs to be in the dimeric form. So in the initial stage, in the G1 phase, it is monomeric. It is having a single unit. So in the A phase, it is generally duplicated, and during the mitosis phase, the centrosome moves away from each other at the equatorial position towards each other. Okay, the opposite position at the equatorial plane, and that is how the centrosome gets organized at the two equatorial plane of the cells, and then the chromosome and the mitotic microtubules are microtubules emerge from this centrosome, and then the chromosomes are aligned with each other, okay, by the help of these microtubules. So at the first stage, the spindle body formation, duplication, and separation occurs, and this is motor dependent. That is, motor proteins that are actin and myotins as well as microtubules play a role in the separation. And then there is a nucleation of the nuclear and cytoplasmic microtubules. Okay, so microtubules are organized on the centromeres, and these nuclear and cytoplasmic microtubules are nucleated from the centromeres, and then these microtubules are attached to the kinetochore. So on the DNA, basically this is the segment of the DNA, and in the between there is a kinetochore, okay, which links the two sister chromatids with each other at the middle phase. And around this is actually called the centromeric region, where the centromere protein is involved. And on the centromere, the kinetochores are attached at the both the sides, okay. And onto this kinetochore, the mitotic spindles are attached. Okay, so the mitotic spindle are emerging from the centromere. So this is the centromere, okay, and this is the mitotic spindle which emerges from it and which attaches to the kinetochore complex. And this is how the DNA remains aligned during the metaphase stage of the cell cycle in the M phase. So microtubule kinetochore association happens and the nuclear envelope also disassembles. Okay, so nuclear envelope which is present in the prophase stage, it gets disassembled so that the chromatin it gets separated into the cytoplasmic region and leads to its segregation. Then the chromosome organization occurs. Okay, and generally during the chromosome organization, basically the sister chromatids cohesions at the centromere and arms facilitate bipolar orientation. So sister chromatids which are cohes with each other in the S phase at the centromeric region, and the arms it facilitates the bipolar orientation. That is, it mediates this bipolar orientation. That is, it has a reverse direction with each other. Okay, during the S phase, and in the S phase, generally during the interphase stage, the chromatins are loosely packed. Okay, are loosely arranged. But at the start of the S phase, due to the protein called cohesin, okay, it start to cohes with each other. Okay, by the two sister chromatids. Between the two sister chromatids, so there is a cohesin protein involved, and in during the S phase. And around the centromere, it helps the binding during around the centromeric region. That is how these two chromated proteins are bounded together with each other. Okay. Earlier, before that, during the replication step, these two sister chromatids are separated, but at the start of the S phase, it is cohes with each other, okay, by the protein known as cohesin. And then the chromosome also condense with each other. So condensation means the tightly packing of the DNA molecule. When during the replication, it is loosely packed, but in by the protein called condensin, chromosome condense the DNA get condensed, okay, and that is how it gets slightly packed with each other. So cohesin and condensin are the two proteins which are important in binding of the chromosomes with each other and in the condensation. So we'll be discussing a little bit detail in the ahead in the lecture, okay? And then there is maturation of the centrosome and kinetochore complex and binding to the microtubules and the congregation of microtubule associated cystochromatic pairs to the equatorial region. Okay, uh, so microtubules are bounded with the centrosome and kinetochore complex, which are present on the DNA molecule at the between the two arms of the chromosome. And then these microtubule associated chromatids are paired with the equatorial region. Okay, the centromeres are present in the equatorial region, and then the microtubule get attached to that sister chromatid and to this centrosome and kinetochore complex. And during the metaphase to anaphase transition, in the metaphase, generally the sister chromatids are bounded by the microtubules. And during the anaphase transition, the sister chromatid gets dissociated and segregated by the dynamic nature of the microtubules. By the dissociation of the microtubules, the sister chromatids are pulled away from each other. Okay, so here in the image, you can see that in the metaphase stage, the sister chromatids are attached in the equatorial plane by the microtubules, which are nucleated from the kineto by the centromere. And during the anaphase stage, the sister chromatids are pulled apart, pulled away from each other by the dynamic nature of the microtubules. And the kinetochore also moves away from each other. The uh, the centromere also moves away from each other, and that is how it mediates the tension and it pulls the centromere chromatids by through each other, okay, in the opposite pole. And in the telophase, generally the nuclear assembly occurs, starts assembling the nucleus, and the centrosomes, the microtubules also get dissociated. And during the cytokinesis, the cells divide with each other, and the chromosomes are again formed in the uncondensed form. Okay, the chromosomes are again uncondensed, and during the interphase, that uncondensed chromosomes uh, are again used for the replication process during the S phase of the cell cycle. Okay, so these are the stages of the mitosis, the different stages, and generally the in the reorganization of actin cytoskeleton. So actin cytoskeleton are helpful during the telophase stage. Okay, which helps in the formation of the contractile ring during the telophase and cytokinesis stage. Okay, so by the contractile ring, this makes this mediates the tension between the two at the cleavage furrow, and that is how the two cells get divided between each other through each other. So this is how the cells occur. Okay, so is it audible to everyone? Right. So this is the different all the events of the cell cycle, and so we'll be discussing in a little bit in detail, like at the different stages of the cell cycle. What are the different proteins which are involved, and how the cell cycle get progress. So this is a general cell cycle division, that is interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. And you can see difference in each of the cell cycle progression. Okay, so as I discussed. So in the prophase, generally the sister chromatids are sister chromatid condense with each other. That is this here you can see in the interphase, the sister chromatids are loosely packed. Okay, and it is not condensed. In the prophase stage, the sister chromatids are found in the condensed form, and the mitotic spindle also gets assembled in the centromere. at the centrosome. So the centrosomes are duplicated during the S phase, and in the prophase, generally the centromeres are present at the equatorial position opposite to each other, and from there the microtubules are organized. Okay, so microtubules are formed from this centrosome. And during the pro-metaphase, the nuclear envelope starts to break so that these chromosomes gets access to this microtubule in the cytoplasm, and it attaches to the centromeric region. It attaches to the kinetochore region of the centrosome of the chromosome. And in the metaphase, generally the chromosomes are aligned with the equatorial plane. Okay, in the between, that is you can see in this diagram. And the microtubules from the opposite spindle pole attaches to this kinetochore. Okay, and it attaches to the sister chromatid. It attaches to the kinetochore of the sister chromatids. So this is how the attachment occurs in the uh, in the in the chromosome. Okay, and in the anaphase, generally the sister chromatids has to pull away from each other. So the sister chromatids separate to form the two daughter chromosomes, and each sister chromatid is pulled towards the spindle pole. Okay, so the sister chromatids are pulled towards the spindle pole, and this force mediates the separation of this spindle pole. Okay, by attachment to the kinetochore complex, and the kineto and the centrosome which are present at the opposite ends also mediates the force such that the centrosome presents pull away from each other. Okay, so this also mediates the force for the microtubules to pull the chromatid apart away from each other, and also the microtubules attached to the kinetochore are also dynamic in nature, so that it assembles and it leads to the disassembly, and then due to the disassembly, the microtubules get shortened. And that is how also this it mediates the pull in the chromatid. Okay, and these chromatids pull away from each other. So this is also the this kind of mechanism happens, which helps in the uh, pulling of chromatid from each other. Okay, from the daughter chromosome. So in the anaphase, generally there are two phases, anaphase A and anaphase B. In the anaphase A, generally the microtubules get shorter and it leads to the cystochromatid separation. And in the anaphase B, the spindle poles which are present in the centrosome are also pulled away from each other. Okay, so that it mediates the force. So this happens in the anaphase B. And in the telophase, the daughter chromosomes reaches to the spindle pole, as you can see. The two daughter chromosomes are present separate to each other, and in the between you can see the contractile ring, okay, which is made up of actin and myosin protein, which helps in division of the cells. And the nuclear envelope, a nuclear membrane start to form again, and and the chromosomes also get decondensed. Okay, so in this form you can see the condensed form of chromosome, but in the telophase stage you can see individual decondensed chromosome. So that during the S phase replication, this decondensed chromosome can be easily accessed by the proteins which helps in the replication complex formation. So that is why the chromosome needs to be decondensed form for the replication stage. And in the cytokinesis, generally the division, which the contractile ring which was formed occurred in the telophase stage. So that mediates this formation of this cleavage furrow between the two cells, and here you can see the separation of the two cells by the contractile ring. And this is called abscission. Okay, so abscission of the cells between the into the two daughter cells. So in the two daughter cells are formed, each containing the one pair of centrioles. Okay, one pair of centrioles are present, and during the S phase, synthesis phase, these pair of centrioles are replicated into another pair. Okay, and that is how this again that pair moves to the another opposite part in the prophase stage. That is how these daughter cells again get divided into another daughter cells. Okay, division into the mitosis. So this is how the cell cycle progression occurs in the M phase. So is it clear to everyone? Hello. Is it clear till now? So is the whether there was a network fluctuation in the between, because my network is not strong over here. So is the voice and the presentation clear? I mean, was there a network fluctuation? You can type in the chat box if you are not. Uh, if there was a network fluctuation in between, you can type in the chat box. Okay, so we will continue from there. So as I told that condensin of the protein, which is required for the chromosome condensation. Okay, and generally the condensin proteins are made up of this subunit, which is known as SMC4 and SMC2, and it has a cape-like structure. Okay, at the bottom, so to this cap H and cap D2 and cap G, and so these condensin proteins are attached by this condensin. Condensin protein attaches to the DNA chromosome. Okay, the chromatids, and then it condenses with each other. So earlier, whether there is a not decondensed chromosome, so this condensin protein binds to this chromosome and helps into this formation of the condensed DNA, so that during the metaphase stage, during the prophase state, the DNA remains condensed, and the shorter DNA will help in the better segregation into the two sister chromatid, okay, compared to the decondensed form, which is a haphazard. And in the by the condensation also, there will be a proper alignment of the DNA molecule in the plane, okay, with the single plane. So this is how condensed protein is there, and here you can see centromeric region. Uh, so DNA, as you know, that DNA is founded by the nucleosome protein. Okay, so these are the nucleosomes, and in the centromeric region, generally the DNA are tightly packed. Okay, this is the centromeric region onto which the kinetochore get attached, and onto the kinetochore the microtubules are attached. Okay, so this is the microtubules. These are the kinetochore plates, that is the inner plate and the outer plate. Okay, and onto the kinetochore the centromeres are attached with the kinetochore. So because the DNAs are completely packed onto the centromeric region, there are high number of nucleosomes present. Okay, on the centromeric region, and that is why the DNA tightly packed, and this is basically inaccessible region of the DNA. In the centromeric region, generally the DNA is inaccessible because of the tightly packed nucleosome complex on the DNA. And in the opposite arms, basically the DNA can be accessible, okay, but not the centromeric region. And it has to be tightly packed because the protein association during the kinetochore formation and the microtubule organization, there will be a multiple number of protein associated with this DNA. So the DNA remains tightly packed in this region. If the DNA, if the kinetochore complex, okay, if the kinetochore is not forming properly, then it will be detrimental during the segregation phase or during the metaphase to anaphase transition. Okay, the chromosomes will not segregate properly, and if the chromosomes are not segregating into equal parts, then there will be a ploidy situation. Okay, there will be an aneuploidy or polyploidy situation in the person, and that will lead to different disease formation. So in the disease formation, you know that there are syndromes known as Klinefelter syndromes and Down syndromes, in which the cells contain one extra pair of chromosomes. Okay, so in the Klinefelter syndrome, basically there is an extra pair of the chromosomes. In the Down syndrome as well, there is a extra pair of chromosomes. Okay, so because of that, there will be a abnormalities present inside the cells and in the human body. So this is why this is critical and important that the centromeric regions and the division during the mitosis stage occurs properly, so that there will not be any complication further. So, so apart from condensing, the condensing helps in the DNA in the one sister chromatid. It helps you to pack the DNA with each other. And another protein is the cohesin, which helps in the binding with the two sister chromatid. Okay, so uh, cohesin basically helps the binding with the two sister chromatids. Okay, and that is how the DNA. So this is the one pair of chromatid, another pair of chromatid, and the cohesin helps in binding between the two. And here you can see here if you find the centromeric region. Okay, and here the kinetochore complex are attached and the microtubules are arranged. During the metaphase, generally between the prophase and up till the metaphase, the chromatids are bind with the cohesin. But during the anaphase stage, the cohesins are degraded by an enzyme known as separase, as we will be seeing. So the separase enzyme will degrade the cohesin, and that is how the two chromatids are pulled away from each other. Okay, so cohesin act as a force between the two, binding between the two sister chromatids. So here you can see that this is the cohesin protein which are bounded between the two sister chromatids, and an enzyme known as separase which is activated and which cleaves this cohesin subunit, that is RAD21. And due to cleavage of this RAD21 subunit, the cohesin does not maintain the binding between two sister chromatids, and that is how it did in the anaphase the sister chromatids pull away from each other. Okay, so earlier the proteins was due to cohesin, the chromatids were remaining intact, but due to the enzyme separase, the bond between these cohesins are broken. Okay, and that is why and by the help of the micro pull from the microtubule as well, by the external pull, the chromatids are move away from each other and separated during the anaphase stage. So this is also an intricate mechanism that during the metaphase to anaphase transition, only the separase will get activated and not earlier. Okay, so if there is a constitution activation of separase, then the chromatids will not be arranged. Uh, two chromatids will not be lined with each other. Okay, by the cohesin protein. So separase is generally acquired by the securine. So this is the securine separase complex, and by the anaphase promoting complex. Okay, that is known as APC, which is activated by CDK. Okay, so APC generally mediates the phosphorylation of securine, and leads to its dissociation by the separase subunit. So now securine is degraded by the APC, and the separase is separated, and that separase actually mediates the cleavage of this RAD21 subunit of the cohesin protein, and that leads to the metaphase to anaphase transition. So when securin is bounded by the separase, then the separase will not remain active. Okay, and the degradation of securin occurs during the anaphase phase only. After that, only the separase will get activated, not earlier. So in summary, this is the stage, that is activation of mitotic CDK brings about the entry into the mitosis, and phosphorylation of proteins by the mitotic CDK, PLK1, that is a polo-like kinase, and Aurora A and B are important for the mitotic event. So this PLK1, PLK, Aurora A and B are also the kinases which are present during the mitosis stage, and it also helps the progression of the mitosis during the metaphase to anaphase transition, and also during the cytokinesis stage. Okay, telophase and the cytokinesis stage by the separation of this chromatid and the formation of the bractyl ring. So these are the different kinases which are also involved. And then there is a spindle assembly, chromosomal reorganization, and bipolar attachment of sister chromatid to the spindle core by the microtubules. Spindle assembly on the kinase centromere, and then the chromosomes get reorganized at the equatorial plane. The sister chromatids are attached to the spindle poles by the microtubules. Well, these are the important events. And condensation and cohesins are also important by restricting the condensation and cohesion of the DNA molecule, chromatid molecule, and then the dissolution of the cohesion during the metaphase to anaphase transition by the enzyme separase. Okay, and cleavage of the cohesin subunit leads to the separation of the two sister chromatids during the mitosis stage. So this is the total in summary detail regarding the how the mitosis occurs. Okay, during the cell cycle. So is it clear to everyone? Is there any doubt or around this? Is it clear to everyone? Can anyone answer? Is there any question from what we discussed currently? If no, then you can also uh, type. Okay, so if you will not respond, then it will be difficult for me to, because I don't know whether uh, the slides are going through fine because of the network connection, and whether you are able to understand or not. Okay, so the purpose of this session is that only that you understand better, because the current session which are ongoing, the recordings, generally it is a recorded session. Okay, so there you cannot interact, and basically if you have any question, then also you cannot, you can ask in the discussion forum, but in this tutorial session you can ask directly, okay, to us, so that your doubts get clear at the initial stage only. That is why I'm going through the lectures as well, the slides which are discussed in the week content, so that if you have any confusion or doubt, so that you can directly ask over there. So you need to respond, okay. Uh, and there is no issue if you on your mic and also talk, because the purpose of this is to learn, okay, and to interact with us. So let's discuss first. We will be seeing the other slides as well, okay. But let currently discuss the certain certain questions first. After that, we will see different proteins which are involved. Okay, so we will discuss about the centrosomes and all the molecular markers. So we talked about the centrosome over here, that the centrosomes plays an important role in the division of this division on the organization of the microtubules and microtubule organization and formation attachment to this microtubules get attached to this chromosome. So we will be talking about the centrosome and the microtubules and the actin and myosin filament over here, okay, which you can say that mitotic spindles are arranged and the contractile ring are also there. So let's discuss some question first, and then we'll be going through other part of the lecture. Okay. So this is the one regarding the mitosis stage of the cell cycle. So you need to respond in this. Okay. So otherwise it will not be fruitful. So you need to respond. So first question is which of the following kinase is not required for mitotic event to take place? These are the model questions. Okay. Previous year assignment question. This is not the current assignment questions. Current assignment is due on 15th, on the next Wednesday. Okay, I think on 21st. So after that only we will discussing that assignment of this week content on the next Saturday class. So these are the example questions which is from these slides. So what will be the correct answer for this? Which is the following kinase is not required for mitotic event? What will be the correct answer for this one? Is it visible to everyone? Is the slide visible to everyone? Can anyone respond? Hello. Is it audible? Are the slides visible to everyone? So this is the first question. We will be solving some questions, okay? Model, some example question. This is the first question. Which of the following kinase is not required for mitotic event to take place? And there are three options: follow-like kinase, Aurora A, Aurora B, and none of the above. So you need to answer which is the correct option. So you can on your microphone and you can answer. So please don't type in the chat box, okay? Because I will not be able to see. Please on your microphone and you can answer what will be the correct option. Option D is correct. Yes. Hello. Is it audible to everyone? So this PLK, Aurora A and Aurora B are the kinases required during the cytokinesis. Okay, so these are the three, as we discussed in the mitosis phase. So these are the three kinases apart from CDK are required. Okay, so not required are none of the above because these all the three are required. So this is the option D is the correct answer. Second is the key event that occurs at the metaphase to anaphase transition is these are the three options: sister chromatid dissociation and segregation, sister chromatid association on the equatorial plane, sister chromatid condensation, and all of the above. So what will be the correct option for this? Please respond, otherwise it will not. Mhm. You can on your microphone and also anyone. What will be the correct option for this one?
All of the above, sir.
Key event that occurs at the metaphase to anaphase transition is all of the above. Others, what do you think? What could be the correct option for this? So sister chromatid dissociation and segregation. So metaphase to anaphase, as we discussed, that dissociation of sister chromatid and the segregation occurs, okay, at that stage. So this one is the correct. Sister chromatid association in the equatorial plane. During the metaphase to anaphase transition, association does not occur. There is a dissociation only. Okay, association occurs at the metaphase stage, not during the metaphase to anaphase transition. This one is incorrect. Sister chromatid condensation occurs during the S phase of the chromosome, okay, during the cell cycle, not during the anaphase stage. So correct option is option A, that is sister chromatid dissociation and segregation. So here you can see during the anaphase stage, the sister chromatid separates and are pulled away from each other, okay. So the option B A is correct, that is sister chromatid dissociation and segregation. The third one is the sister chromatid cohesion is established in the which phase? So we discussed about the protein known as cohesin, which helps in the uh, binding of the two chromatids, okay, to form to form a pair of chromosome. So you need to select the option at which stage does it occurs. That is during the G1 phase, S phase, metaphase, or the anaphase. So in the S phase, generally the sister chromatic cohesion occurs. Okay. Option B is correct. So this is another question. Which of the following class of protein are not present in the pericentral material? Generally, the pericentral we'll be discussing that the pericentral are the material. Pericentral is present inside the centrosome. Okay. Centrioles are there, and centrioles are covered by this pericentral material, in which there are many cell cycle regulator proteins and all the other proteins which help in the division process. So the correct option is none of the above. Okay, because are not present. So this all the three molecules, that is cell cycle regulators, signaling molecules, and the microtubule organizers are present in the pericentral material. So here you can see that the centrosome we discussed. Okay, the centrosome. Earlier we discussed that centrosome uh, contains an organizing center. That is, from there the microtubules are arranged from there. Okay, and during the S phase of the cell cycle, the centrosomes, here you can see centrosomes divide, okay, inside the pericentral material. So the centrosomes are covered by this pericentral material. And in the pericentral material, there are the cell cycle regulators, signaling molecules, microtubule organizers, and that are collectively called the microtubule organizing centers. Okay, so this whole complex is called MTOC, that is microtubule organizing center. And during the S phase, the centrioles get divided, and in the late S and G2 phase, there is complete replication of the centriole molecule. And during the early prophase, the two centrioles will move apart from each other so that it arranges at the equatorial plane at the opposite direction. And that is how the centrosome gets divided. Okay, but because during the after the division, there is only a one pair of centriole present, and the centriole need to be present into two pairs so that the microtubule get arranged from the opposite end of the two support the sides. That division occurs in the S phase of the cell cycle. In the prophase, generally it is pulled away from each other. So pericentral materials are the material which are embedded inside the center. The cell cycle regulators, signaling molecule, and microtubule organizers are present in the pericentral material. So microtubules that overlap with each other when in antiparal orientation are called. These are the four kinds of microtubule: astral microtubule, kinetochore, interpolar microtubule and intrapolar microtubule. So if we have seen the this week lectures, then it was about the microtubules. It was discussed. Just a sec. So what would be the correct option for this? The option C is correct. Interpolar microtubules. Okay, so microtubule that overlaps with each other when in antiparallel orientation are called interpolar microtubules. And the interpolar microtubules, generally there are three classes of the microtubules. That is, one is astral microtubule, kinetochore, and interpolar. So astral microtubules are present in the centromeric region at the pericentral material, where the centriole are there in the opposite direction. Okay, this astral microtubule generally helps in the maintaining the polarity, so that it attaches with this membrane of the cell and it mediates the polarity, the localization of the centromere at the equatorial plane. So these are the astral microtubules, which are formed in the opposite direction of the chromosomes which are present. The kinetochore microtubules are the microtubules which are present attached to the kinetochore region of the chromosome, and the interpolar microtubules are microtubules which are not attached to the kinetochore and which overlaps with each other over here. Okay, you can see. So these are the interpolar microtubules which overlaps with each other. So these are the three kinds of microtubules, and here you can see that microtubules overlap each other in antiparallel orientation because these are converging, emerging from the opposite poles, that is interpolar microtubule. Which of the following protein is necessary for metaphase to anaphase transition? These are the four kinds of protein: helicase, separase, glutamidase, and phosphatase. During the metaphase to anaphase transition, which of the protein is essential? So you need to answer this. We discussed about this.
Separate, sir.
Option B is correct. Separase generally helps in the cleavage of this condensed cohesion unit. Okay, red 21, and that leads to the separation of the chromosome during the metaphase to anaphase transition. So separase is the correct option. The spindle body present in the budding is equivalent to which organ list? So the spindle body in the budding is, so this we haven't discussed currently. So if you have gone to the week, so that it would discuss. Correct option is the centrosome. Okay, so in the Saccharomyces cerevisiae, basically the cytokinesis occurs in this stage, such as this is the chromosome. This is the centromere, and this is the septine ring. So in the mammals generally the centrosomes are divided. Okay, the cells are divided, and then there is a septine ring formation by the actin and myosin. But in the Saccharomyces cerevisiae, The septin rings are formed before the division, okay, in the G1 phase itself, so that it signals that at this stage the division will occur. And during the G1 stage, the septin ring is formed. During the S phase, the chromosome, as you can see, it is replicated, and the centromeres are divided, as found in the mammals as well. And there you can see the septin ring are growing, okay, compared to the previous. Septin ring are growing, and from there the budding will occur. Second one is cervix is the budding is. So from there the budding will occur, and that actually signals that the division will occur at this end. Okay, septin collar will form, and at the metaphase the chromosomes, the centromeres are the centrioles are present at the opposite pole, and it attaches to this kinetochore region. And during the anaphase, the two chromatids are separated through this myosin ring. Okay, and later at the end of the mitotic, the septin splitting will occur, and the two cells will be divided from the mother cell. So that is how the division occurs in the Saccharomyces cerevisiae. And the spindle body present. So this is the spindle body is formed, and the spindle body in the body is equivalent to centrosome. Okay, uh, which signals the division at this stage. So that is equivalent to the centrosome present in the humans, in the mammals. So now this is regarding the FRAP experiment. The FRAP experiment on the microtubules yields which of the following inferences. So individual microtubules are dynamic and the subunits are constantly being replaced. Individual microtubules are constantly polymerizing at both ends. Tubulin remains at the equatorial plate of the cell cycle, and the microtubules are static and stable in nature. So what do you know about FRAP? What is the FRAP technique? Can anyone tell? Like, is anyone aware what is FRAP? FRAP is a technique. Okay, so you can tell the full form first. What will be the full form of this FRAP? Fluorescence recovery after photo bleaching. Okay, so in this technique, basically, photo bleaching is done, and the fluorescent dye is used, and by the photo bleaching. So first, the fluorescent dye will be used. Okay, to label a certain protein. For example, let's take an example of the plasma membrane. Okay, general example. Plasma membrane, as you know, that they are dynamic in nature. Okay, so how can one prove that the plasma membrane is dynamic? So first, in the plasma membrane, you generally label certain part of the plasma membrane by some dye. Okay, and then you bleach that dye. Okay, you completely label the plasma membrane by certain dye. Okay, and then you by the radiation you provide the photo bleaching. So at the place where the radiations are radiations are passed onto the plasma membrane, at that place the fluorescence will not get emitted. Okay, but if the plasma membrane is dynamic, then this bleach region will be replaced by the other part of the plasma membrane which are fluorescent. So after a certain point of time, photo bleach regions will be replaced by the fluorescence region again, which is labeled. Okay, that suggests that it is a dynamic in nature, such that bleach region will be moved away, and the fluorescent region will take its place. So that suggests a dynamic property of the plasma membrane in general, as an example. If you want to prove a certain molecule that it is dynamic in nature, the process dynamic, then you use the this technique called as fluorescence reactivation after photobleaching. So by this experiment, one can prove that the microtubules are dynamic and their subunits are constantly being replaced. So for example, if you label the microtubules by their certain dye, okay, and you do the photo bleaching or in the specific region, and after a certain point of time, you observe the fluorescence. Then you see that the photo bleach region are again fluorescing. So then you can speculate that. Because of the dynamic nature of the microtubule, the photobleach region are being replaced by the adjacent regions in the microtubule. So option A is correct in this, that the Frap experiment generally dictates the dynamic nature of the microtubules. And generally the dynamic nature of microtubules, we already discussed that it helps in the pulling of the microtubules. Okay, the microtubules needs to be dynamic during the metaphase to anaphase stage. So it at the during from the prophase to the metaphase stage, it continuously gets formed so that it attaches to the chromosome. And during the anaphase stage, it continuously depolymerizes. Okay, so because of the depolymerization, the microtubules get shorter and shorter, and that is how the two chromatids pull away from each other. So microtubule needs to be dynamic during separation and segregation phase, so that the chromosomes are separated from each other. Okay, so option A is the correct option. So then these are the three statements regarding the spindle attachment to this chromosome. The spindle attachment, that is the microtubule attachment to the chromosome. So these are the three statements. So after this statement, you need to choose the correct option. What will be the correct option in this case? The first one is the sister kinetochore are assembled in back-to-back orientation to reduce the chance of mono-orientation. Okay, and cohesion in the centromere makes the paired sister chromatin to be more fluid in nature. Pi orientation of chromosome is important to generate tension at the kinetochore between the sister chromatin. So let's look one by one option. Sister kinetochores are assembled in back-to-back orientation. So kinetochore, as I told, that are assembled on the centromeric region, and they are arranged in the back-to-back orientation, okay, at the opposite orientation, so that it reduces the chance of mono orientation. So there are two sister chromatids. These are two sister chromatids, and around that centromere are attached. And over there, this is the orientation of the kinetochore. So they are attached into opposite direction with each other, okay, so that the two microtubules will attach. So if the two kinetochore will attach at the same end rather than the opposite direction, then the microtubule is attached at these two ends, okay. And if there is no kinetochore present in this end, then microtubule will not attach over here, and that will lead to the uh, not separation of the chromosome will separate at one end only, okay, at both the pairs. The sister chromatid will not separate it equally. So the sister kinetochores need to be assembled in the opposite orientation, back-to-back orientation, okay. Then because in place of the mono orientation. So this option is correct. Cohesion at the centromere makes the paired sister chromatid be more fluid in nature. So this suggests that cohesion at the centromere. So this is the centromere region, and the cohesion, which is mediated by the cohesin proteins, makes the sister chromatid to be more fluid in nature. So this option is actually incorrect, because if the cohesins are there, okay, cohesin actually helps to be more rigid rather than the fluid. So because of the cohesin protein, the chromatin regions are rigid in nature. Okay, it does not become fluid. In fluid region, basically, is mediated by the degradation of cohesin. Okay, that will allow the cystochromatic separation and uh, formation of the euchromatin region. Okay, the loosened part of the chromosome, which is found in interface. So this option is incorrect. Bi-orientation of chromosome is important to generate tension at the kinetochore between the sister chromatids. So bi-orientation of chromosome is important to generate tension at the kinetochore. So this is option is actually correct, because bi-orientation of chromosome, that is chromosomes are present in the dimeric form. Okay, so over there the kinetochore will remain attached in the opposite direction, and then it will microtubule will attach at the kinetochore and will pull away the sister chromatids between each other. Okay, and produce it by producing the tension. So the bi-orientation of the chromosome is essential rather than the mono-orientation, as I discussed over here, that the sister chromatids are not oriented in the duplicate form, then the then the tensile will not form properly, okay, between the two sister chromatids. So option 1 and 3 is correct. That is option C. So is it clear, this part? This part, is it clear to everyone? Am I audible? So here you can see an example, okay, kinetophore plates which are rated to the microtubules, and the bar orientation actually helps in the proper segregation of the microtubule. So now we will discussing more about the week 3 assignment, okay, previous week assignment. So you had submitted this assignment on Wednesday. So you might, if you have any doubt, then you can, in any of the questions that you can ask over here. So this was the first question. Pick the incorrect statement about the S phase of the cell cycle. So DNA duplication occurs in this phase. Once the cell crosses the restriction point, it enters the S phase. Certain stimulus can reverse the progression even of the cell that cross the restriction point. And in yeast through S phase requires CLB5 and CLB6 cyclins. So these are the four options, and you need to pick the incorrect option out of this. So what will be the incorrect option out of this? Incorrect statement out of these three. Option C is incorrect. First option is DNA duplication occurs in this phase. So in S phase, intelligent DNA duplication occurs. Okay, and once the cell crosses the restriction point, it irreversibly enters the S phase. So we had discussed earlier that if the G1 phase is completed at the restriction point, okay, the cell enters into the S phase. It cannot be reversible once the cell crosses this restriction point in the G1 phase. Okay, in E's progression through S phase requires the CLB5 and CLB6 cyclid. So this is the correct option. But CLB5 and 6 are required during the S phase. Okay, these are the cyclins which are present. In mammals, generally, there are cyclin E complex, okay, by the CDK2 cyclin E complex in the S phase, but in yeast there are CLB5 and 6 cyclin. And certain stimulus can reverse the progression of S phase even though the cells are close to the restriction point. So this is incorrect because once the cell has passed through the S phase, it cannot be reversed. Okay, so option C is incorrect. So the origin recognition complex binds the replication origin at which stages of the cell cycle. So you need to answer this one. At late G1, throughout the cell cycle, at early S phase, or throughout the replication. Which is the correct option out of this?
Throughout the cell cycles.
So option B is correct. Throughout the cell cycle. So here we discussed about the DNA replication and the origin replication complex. Okay. So that origin recognition complex is bound to the origin, and it serves as a launching pad for the additional proteins over there. But in the this origin, this ORCs are present at the throughout the cell cycles of the throughout the cell cycle. Okay, ORCs are bounded throughout the cell cycle, but when it is assembled by the CDC6 and CDT1 protein and recruitment of the FCM helicase, then it forms a pre-replicative complex, and this pre-replicative complex are only found during the early G1 at the late G1 phase. Okay, so this is how the replication then occurs during the S phase of the cell cycle. This is the third question. Which of the following is incorrect about the DNA replication? Primer template junction is necessary for DNA replication to begin. The extension of the primer occurs at its 5' end. DNA polymerase is required to catalyze the formation of the phosphodiester bond, and the correct base insertion is ensured by complementary base pairing with the template. So these are the four options, and you need to select the incorrect option. Which of the option is incorrect in this case?
Second option.
Yeah. So option B is incorrect. The primer template junction is necessary for DNA replication to begin. Okay, so this is correct. It requires a primer and template junction. DNA polymer is required to catalyze the formation of phosphodiester bond, so this is also correct. And the correct base insertion is ensured by complementary base pairing with the template. So correct base insertion is actually ensured by the complementary base pairing. That if the template is correct, then only the correct base pair will occur. So that is why it requires a template during the DNA replication. So these three are correct. The extension of the primer occurs at 5' end. So DNA replication, we know that it occurs from the 3' end, okay, not the 5'. So this is incorrect. The phosphodiester bond is formed between the which of these molecule, how it is formed. So you need to select out of these four what is the correct option. The DNA replication we discussed regarding this. So option A is correct. 3 days OH of the preceding base of the alpha phosphate of the 5' base. So here you can see that this is a one nucleotide subunit, and this is the incoming nucleotide. Okay, so at the 3' end of the ribose sugar of the current nucleotide of the current preceding base, the phosphodiester bond is formed by the incoming 5 days phosphate of the incoming nucleotide. Okay, the 5' phosphate which are attached to the ribose sugar at the fifth position, with carbon position, makes a phosphodiester bond by the at the third carbon position of the ribose sugar of the already present nucleotide. So this is the option is correct. 3 days OH of the preceding base to the alpha phosphate of the 5' of the following base. Option is correct. So Okazaki fragments, we already know what are Okazaki fragments. So what will be the correct option in this case? Okazaki fragments are Okazaki fragments are short segments of DNA strand made of 100 to 2000 nucleotides. Okay, so these are the Okazaki fragments basically, which are extended from the primers while the lagging strand. That are generally it varies from the prokaryotes and the eukaryotes. In the prokaryotes, generally it is 100 to 400 nucleotides. In the eukaryotes, that is 1000 to 2000 nucleotide base pair. So overall, it is 100 to 2000 nucleotide of fragments. So the prokaryotic DNA replication. The prokaryotic chromosome is usually circular. Prokaryotic chromosome multiple origins. Replication initiation requires initiator protein and primosome, and the replication proceeds bidirectionally. So out of these four options, you need to select one with the incorrect. What is the incorrect statement out of this? Is it audible? Hello. So option B is incorrect over here. The prokaryotic chromosome multiple origins. Prokaryotic have a single origin or multiple origin. Okay. The prokaryotic chromosome is usually circular. It is correct. Replication initiation requires initiator protein and primosome. So the replication initiator requires the primosome and initiator protein. And the replication generally occurs in the bidirectionally, okay, in the prokaryotes as well as the eukaryotes as well. Option B is incorrect. So Kazaki fragments are formed in which strand during the DNA synthesis? The leading, lagging, neither leading or lagging, none of the above. So which can be the correct option in this case? So Kazaki fragments are formed in the lagging strand. Okay. The option 8, the unwinding of the parental DNA helix during the replication is carried out by the replication DNA helicase called the unwinding of the parental DNA during the replication is carried out by the DNA helicase called. Which one of the option is correct in this case?
Option A.
DNA helicase. Option B is correct. DNA B. DNA A is actually the origin recognition and it melts the AT region. Okay, that is an origin recognition initiator protein. It is not an helicase. The helicase is DNA B, which unwinds the DNA at the replication fork. Yes, option B is correct in this case. Pick the incorrect statement about the eukaryotic DNA replication. Eukaryotic chromosomes are linear. They have multiple origin of replication. All the origin fire simultaneously, and DNA replication happens only once per cell cycle. So which of these is incorrect?
All the origins fire simultaneously.
No. The option C is incorrect because in the eukaryotic generally origins does not fire simultaneously. It occurs in phase wise. Okay, so certain origins will fire early, certain origins will fire late. So this option is incorrect. Others are option are correct. And this is the last question. In yeast, the leading strand is synthesized by which polymerase? So there are four different polymerases: pol B, pol beta, delta, alpha, and epsilon. So generally, pol epsilon is mediates the leading strand synthesis in the Saccharomyces cerevisiae. Okay. In the E. coli, generally the pol 3-4 complex is there, but in the eukaryotic system, pol delta epsilon mediates the leading strand synthesis, and pol delta mediates the lagging strand synthesis. So are these questions clear to everyone? Are these questions clear to everyone? Is there any doubt regarding this question? So we will discuss about some more aspect of the centrosomes and all. Okay. So centrosomes, as we discussed, that it has it is a body from which the microtubules are organized. Okay, and these microtubules are attached to this kinetochore region of the chromatid. So we'll be discussing about the centrosome. The centrosome is a structure that modulates the processes such as proper mitotic spindle placement and cilium formation. Okay, and centrosome is composed of two barrel-like structures called centrioles. So these are the centrioles. The whole unit is called centrosome. Inside it, there are centrioles present, which is a microtubule unit. So individual microtubules units make up the centrioles. These centrioles are organized into the pericentral material. So material which is covering the centrioles are called the pericentral materials, and it doesn't have any membrane. Centrosome replication, as we discussed earlier, that during the G1 phase, the cell contains only one pair of centrosome. But during the S phase, because in the mitosis phase, there should be a two pair of centrosome. Okay, so the duplication occurs in the S phase. Here you can see each centrosome are duplicating one pair of one daughter centrioles. And in the late G2 phase, late S and G2 phase, these centrioles are duplicated completely inside the one pericentral material. Okay, one centrosome material that is called microtubule organizing center. And during the early prophase, these centrioles divide and transfers to the opposite fold. And from that, the microtubules will be arranging during the late prophase and the metaphase stage. This is how the centrosome are arranged. So in this diagram, you can see that this is the interphase chromosome, and in the G1, here you can see the centrosome, dividing centrosome. In the late G2 phase, it is completely divided. In the early prophase, the centrosomes are translocating to the opposite folds, and the chromosomes are also decondensed form during the early prophase. In the prophase, the centrosomes are located at the equatorial position at the folds of the nucleus, at the folds of the cell, and the DNA are also condensed in the condensed form, and the nuclear membrane start to disappear. Okay, start disintegrating. In the metaphase stage, the microtubules are attached to the kinetochore region of this centrosome of this chromosome, and in the anaphase, the microtubules start pulling the sister chromatids apart from each other by attaching to this kinetochore and involving the dynamic nature of the microtubules. And in the telophase, generally the contractile ring is formed, and the sister chromatids are separated into individual nucleus. Okay, and the chromatids are again converted into chromatin. Okay, loosely packed chromatin region. And in the cytokinesis, due to formation of cleavage furrow, this separation occurs. Okay, into the two daughter cells. This is the examples of the uh, this is in general the overall stage of the cytoskeletal development. So mitotic cytoskeletal machines are like mitotic spindle and the contractile ring present. Okay, and microtubules are present in this case at by attachment to these microtubules, and the actin and myosin's filament help in the formation of contractile ring around the microtubule, around the cells. Just a minute. Okay, so generally these are the two mitotic cytoskeletal machines present during the cell division. Okay, one is the microtubules and another is actin and myosin filaments. And uh, during the mitotic spindle formation, mitotic microtubules are form the mitotic spindle. And actin and myosin helps in the contractile ring formation. Okay, during the cell division. So as we discussed, the mitotic spindle, there are three classes of the microtubules. So one is astral, another is kinetochore, and third one is interpolar. So astral microtubules are present at the equatorial position, okay, which are in the opposite direction of the kinetochore microtubules. And this astral microtubules actually helps in the polar polarization of this kind of this centrosome, okay, by mediating the polarity to the centrosome. Kinetochore microtubules are attached to this kinetochore region, and interpolar microtubules are attached in the interpolar region between the centromere. Okay, so these are the three kinds of mitotic spindle. So in general, microtubules are filaments that extend through the cytoplasm, and there are two subunits in the microtubules. One is the alpha, and another is beta tubulin. So microtubules are formed by alpha and beta tubulin subunits, and microtubules provide track for the movement of these two motor proteins, that is kinesin and dynein. So kinesin and dyneins are the motor proteins which help in the movement through the microtubules and helps to deliver cargo at the specific location. The microtubules are attached to the sister chromatid at the centromere via kinetochore. And there are three kinds of microtubules. One is interpolar, another is kinetochore microtubules, and third one is astral microtubule. So all these three microtubules play a role in the mitosis. Okay, and it helps in the division process. So this is the microtubule organization, and here you can see the alpha and beta tubulin. Uh, this is protofilament, one protofilament, and 13 total protofilament makes up this cylindrical microtubule sector. Okay, so 13 filaments makes up the total microtubule, and each protofilament is made up of alpha and beta tubulin. The light blue portion is the light blue colored is the alpha tubulin, and the light dark blue is the beta tubulin. So this alpha and beta tubulin actually mediates a different process that alpha tubulin binds GTP quite tightly. Okay, and in the beta tubulin generally the GTP is not bound tightly. And if the GTP is converted, GTP is generally hydrolyzed into the GTP molecule. And if the microtubules are converted, GTP is converted to GTP, so that subunit tends to dissociate. Okay, so generally, if the microtubules are exposed to the beta tubulin, then the microtubule will get dissociated. But if it is exposed to the alpha tubulin, the microtubule will not get dissociated. Okay, so this is the dynamic property of the microtubule. So the polymerization generally occurs at the plus end of the microtubule. So plus end of the microtubule will have more of the alpha tubulin, okay, which provides the capping. So if it is at the GTP, then it will provide the capping at that microtubule position. Plus, cellular. Okay, and general inhibitors such as this Paxol, Metronidazole, and colchicine, it binds and stabilizes this microtubule at the at its cap region so that it doesn't lend to polymerization. Okay, so this is the K inhibitors, and this is the like orientation so that when it is GTP bound, that is alpha is there, then it forces the protofilament into intact form. But if the GTP is lost, okay, if it is exposed to the GTP form of the microtubule, a within subunit, then it destabilizes the microtubule and microtubule tend to depolymerize. So this is the dynamic nature of the microtubule depending on it. And the half-life of the microtubule, so the FRAP experiment, which we discussed, that fluorescence recovery after protobleaching, that in metaphase generally, when you provide the laser beam, you bleach that for microtubules. So at what time it is getting recovered, so that depends dynamics. So if it is highly dynamic, then it will recover in a shorter period of time. But if it is less dynamic, then it will recover at the half-life will be very high. So the experiment was performed, and here you can see that in interface, generally the half time in which the microtubule get recovered was 5 minutes. But in the metaphase stage, the half-life was 15 seconds. Okay, so the dynamic property, the dynamicity was higher in the metaphase. So this dynamicity actually suggests that in the metaphase, because of the mitotic spindle assembly and disassembly, because of the segregation of the chromosome, the microtubules are highly dynamic at that stage. Okay, when metaphase compared to the interface stage, where the microtubules are not present. Okay, so these are the so this is in general regarding the mitotic checkpoints, okay, and the complete stage of the mitosis. First is the spindle assembly checkpoint, so that it ensures that the cell do not enter the anaphase. Another bi-oriented at the spindle, okay, and activation of the CDC20 and APC actually helps in the spindle assembly formation. So if the kinetochore is not attached by this mitotic spindle, that signals this CDC20 APC activation, and it provides a negative signal to this CDC20 and APC activation and blocks this mitotic metaphase to anaphase transition. And that is how these checkpoints might mediate, okay, if the mitotic spindle is not attached to the kinetochore. And when the kinetochore is attached by the mitotic spindle, that triggers the metaphase to anaphase transition. And generally, the APC, anaphase promoting complex, is activated, which is activates by binding to the CDC20, which is a cyclin dependent kinase. CDC20 helps in the activation of this mitotic CDK, and that will help in the promotion to metaphase into anaphase complex. And also the activation of securin. Okay, APC, anaphase promoting complex, also targets the securin protein and mediates its proteolysis. So as I told that securin is bounded by the separase, and the APC generally targets the securin and mediates the dissolution so that the separase become active, and separase generally cleaves the cohesin and condensing protein, cohesin protein importantly, okay, and that is helping the separation. So separation is essential, that is mediated by the separase, and separase is activated by the APC complex. And APC is generally activated at the metaphase to anaphase transition by the CDK cyclin complex. So that is how the the cells regulate the processes. And there are also other molecular motors such as kinesins and dyneins, which are helpful in the which moves along to microtubules and help in the deliverment of cargo. So there are depending on the depending on molecular method depending on the carboid which is inside. So type of filament that they bind, whether it binds the actin and microtubules, that way it can be classified. Direction in which they move along the filament, that is the plus and or minus n direction. For the cargo they carry, that is the cytoplasmic filament or the membrane bone. So the molecular motors, so they can be classified depending on all the three types. Okay. In the head region generally, uh, the head region are the motor domains and it converts the ATP into ADP. So ATP hydrolysis helps in the movement of this molecular protein to on the microtubules and the microfilaments. Okay, so it mediates the movement through the ATP hydrolysis. And these are the two kinds of different kinds of motor proteins, such as myosin are generally founded to the actin. Okay, myosin family binds the actin filament, and kinesins are dyneins bind to the microtubule filaments. Okay, so these are the three different classes of the microtubules. Okay, so these are the depending, and here you can see the actin and myosin contractility. So in budding is generally the septum rings are formed at the phase, okay, by the actin and myosin. So actin, the the monomeric region is globular actin, and by the help of this forming proteins, it is converted into F actin. Okay, and this F actin is bounded by the myosin protein, and that is helping the contractility by the myosin and actin complex. So here you can see the myosin and actin complex are formed during the cleavage furrows, and which helps in the separation of these two cells. Okay, in the budding yeast as well, here you can do the septin ring formation. So this we discussed the septin ring formation, dealing with the cells decide the location through which the the cells into divide. That is formed by the actin and myosin protein, and through which the cells segregate through each other. Okay, during this phase for the cytokinase. So this was regarding the process of mitosis and the different phases of the mitosis: prophase, metaphase, anaphase, telophase, and cytokinesis. And we discussed about the regulation based on that, okay, by condensing and cohesing protein importance, and the separase, what how the separase function. We also discussed some assignment questions. So if you have any doubt, then you can ask from this class lecture, and if you don't have any doubt, then we can end this session. So do you have any doubt regarding this, what we discussed? Okay, so if you don't have any doubt, then we can end this session, and thank you all for joining.